Assistant Professor Dr. Marwa Hamid Al-Khafaji. The subject is Food Microbiology for the 4th class students in the Department of Biology, College of Science, University of Baghdad. Our 4th lecture is Microbial Spoilage of the Food. Microbial spoilage is any organoleptic change in food makes it unacceptable to consumer. The change usually occurs in the flavor, color, odor, taste, and texture. In spite of differences between signs of spoilage according to the type of food, there are 3 main signs of microbial spoilage are common in most of foods. Number 1, bacterial discoloration. This table shows the appeared color on the food and the causative agent. First type is fluorescent yellow-green color. It's caused by Pseudomonas fluorescens due to the production of pyoverdine pigment. The second color is a blue color produced by Pseudomonas aeruginosa due to biocyanine pigment. These pigments are extracellular pigments, as we'll see in the next slide. Black color may be appeared on food due to the growth of Bacillus species and the production of melanin pigment. Yellow color by Micrococcus luteus or Flavobacterium or any other bacteria produce yellow color. Rose by Micrococcus roseus. This microorganism always related to equipment contamination during milk collection. Red color by Serratia maricens. The figure shows Pseudomonas aeruginosa, which produce a blue color on milk agar. As we see, it is extracellular pigment that produced in the media, not only restricted to colonies. This figure shows Pseudomonas fluorescens, also an extracellular pigment, fluorescein. The figure shows a piece of meat, and we see here the black color by melanin. The causative agent is Bacillus. This figure shows Micrococcus roseus, pink color or rose, and this Micrococcus luteus with yellow color. This is Serratia maricens on a piece of toast. While this figure, what I'm proud of it's mine. When I search about these figures, I found my own figure in the media about Staphylococcus chromogenes cultured on milk agar. Not only bacteria could cause discoloration of food, and this type of spoilage may be due to yeast discoloration, which is either white pale colonies of non-pigmented yeast like Saccharomyces cerevisiae, or red pink colonies of pigmented yeast as Rhodotorula. Molds also caused discoloration. The molds are widely distributed causative agent of food spoilage, produce wide range of water-soluble and insoluble pigments, and this color mold colonies on food surfaces. The figure show strawberry with geotrichum, as I think, white mold, and a piece of bread or toast covered with, from the first look, it looks like Aspergillus flavus, and a piece of cheese covered with penicillium, green bluish. The second type of food spoilage is a slimness. Slimness may be two types. The first type is the production of light slim by pectolytic bacteria, and this is for plant origin foods, when bacteria of pectolytic type like Erwinia carotivora, which lyses the pectin in plant tissue. Pectin is an adhesive material between plant cells. The figure shows carrot with a light slim. The second type of slimness is the production of a heavy viscous slim. You may know that this is a production of viscous polysaccharide on the food, such as levain or dextran, from food mono or disaccharides. So, the microbes such as Pseudomonas viscosa or Leuconostic mesenteroids, these microorganisms contaminate food like cheese or meat and turn the food mono or disaccharide to a heavy viscous polysaccharide like levain or dextran. The third type of food spoilage. The first was discoloration, the second was slimness, and this is the third type of food spoilage, which is changes in flavors and odors of the food. The normal acceptable flavor of the food may be changed to non-acceptable flavor in a spoiled food. The change occurred by two mechanisms. The first mechanism is microbial destruction of natural flavor compounds of the food, as in destruction of natural flavor of butter, diacetyl, to off-flavor acetyl methylcarbinol by the microorganism Pseudomonas fragi, or destruction of natural fruit organic acid to unacceptable alcohol ethers, ketones, or aldehydes. The second mechanism to change the flavor or odors of the food is by microbial production of unacceptable off flavors or off odors in the food, as the following examples in this table. So we'll show the off flavor produced by a causing microorganism and the mechanism of production, which is followed by these microorganisms to produce these off flavors or odors. The first one is souring flavor, may be caused by Lactobacillus acetobacter, Propionibacterium, Clostridium butyricum. How could these bacteria cause souring flavor? By using food sugar and converting it to lactic acid by Lactobacillus, or acetic acid by Acetobacter. Propionibacterium produce propionic acid, while Clostridium butyricum produce butyric acid. Bitterness flavor caused by Pseudomonas or Bacillus. The mechanism of Pseudomonas or Bacillus to produce bitterness flavor on food is by protein hydrolysis, fatty acid degradation, or sugar bitter aldonic acid. Sulfide odor caused by Clostridium or Proteus. The mechanism to produce sulfide odor by Clostridium or Proteus is by converting a protein to sulfuric amino acid and H2S. Rancidity caused by lipolytic microorganism. The mechanism used by these microorganisms is by consuming lipid to fatty acid and ketone, and a clean flavor by a heavy growth of E. coli, which consume tryptophan to scatol and indole. Earthiness flavor, soil flavor, by streptomyces, production of geosmin. This geosmin absorbed by food contaminated with soil. Cabbage and potato flavor by Pseudomonas due to the production of vegetable and fruit flavor in a refrigerated milk or meat. Soapiness of meat and cheese by Pseudomonas due to reaction between free amino acids and free fatty acids lead to production of soapiness compounds. So when Pseudomonas lyses the protein and produce a free amino acid and lyses the fat of the meat and produce fatty acid, the reaction between these free acid and a free amino acid and the free fatty acid lead to production of soapiness compounds. Our next subject is about indicator bacteria of food contamination, and the microbiological standard of the food. Regarding indicator bacteria, there are three purposes for which we should examine the food and food plant for contamination indicators. The purposes are to detect the food is healthy or not healthy for human consumption, to assist the sanitary practices of food plant, and to detect if the official standards are meeting the produced food for compatibility with official standards and sanitary constructions of this country, of each country. There are reasons behind why we must search for indicator bacteria of contamination of the food, not for the pathogenic bacteria. Number one, in most cases, such as infectious hepatitis, detecting methods for causative agent, which is hepatitis virus in this case, are not available to food microbiologists. So you remember that when a person gets infected with hepatitis virus, we take his serum and make the test searching about antibodies for the virus. So there is no kit for food microbiologists that detecting the hepatitis virus in the food. For this, we can't detect hepatitis virus in food samples. The second reason for other foodborne infections, such as shigellosis caused by Shigella dysentery, the methods may be unreliable, especially when the pathogen distributed in food heavily contaminated with other microorganisms. So we can detect the small numbers of Shigella in food heavily contaminated with E. coli or staph or salmonella. While this food may cause shigellosis to healthy one when he consume it, but it's still undetectable. Test for pathogens directly, very dangerous to lab workers. This is the third purpose or reason behind searching about indicator bacteria rather than pathogenic bacteria. For these reasons and difficulties, all microbiological lab firstly test species in the food, species which are more readily cultivated, microbial species which are readily cultivated, enumerated, not dangerous to workers and environment, not toxigenic, normal inhabitant of human and animal intestine, which indicate the exposure of food to waste and conditions containing pathogenic microorganisms and their toxic products. These species used are called indicator bacteria. So, indicator bacteria have characteristics which is readily cultivated, enumerated, not dangerous to workers nor to environment, and it must be normal inhabitant of human and animal intestine. Such indicators also help in assessing the quality of the food and the presence of dirty particles such as insect parts, vermines, hair, rodent excretion, etc. At least each country have a microbiological standard of the food and drinking water, if not each city. This microbiological standard give assurance of that the food is acceptable to consumer from a public health standpoint, and the food is acceptable to consumer from an aesthetic viewpoint. The food product must be with high nutritive value and normal characteristics, and the food must have a keeping quality that should be expected. This is what assured by microbiological standard of the food. You are as a microbiologist. You may have to issue a food microbiological standard for your country. So it is important to take in consideration 5 principles to initiate a food standard. The first principle is determination of the higher limit of microorganisms permitted in each food. Principle number 2, determination of the types and higher limit of each type of microorganism in each food type. Principle number 3, all types of food must be free of any pathogenic and toxigenic microorganisms, in some cases except Staphylococci. Principle number 4, for the same food may be more than one standard according to the physical state of the food: fresh, canned, minced, dried, frozen. So each state of the food have a special standard. The principle number 5, in some cases, as in the milk, food may be divided into many grades, A, B, C, according to the microbial content, and a standard for each grade, for grade A, B, and C. The 6th principle is the standard may be not identical for all countries, all towns, because different conditions between these countries as the following: level of food productivity and available foods. So in some countries they depending on seafood, so they have a special standard for seafood. B, economic situation. When a poor country, they have no choice to permit some types of food and prevent some types or grades. So it will be a low standard for poor countries. The sanitary situation also. The nutrition habits. Nutrition habits, like we burn food when we cooking, while other countries may eat fresh food like raw meat without cooking or half cooking. So this nutrition habit also affect the standards. Emergencies, outbreaks such as war, catastrophes also affect the implementation of standards, food standards. So it will be low standard in these cases. There are organizations responsible for microbiological standards of the food. We are, as a microbiologists, work in these organizations to issue food standards. WHO, World Health Organization, FAO, Food Agriculture Organization, CAC, Codex Alimentarius Commission, FDA, Food and Drug Administration, APHA, American Public Health Association, EPA, Environmental Protection Agency, EOF, European Organization of the Food, COSQC, Central Organization for Standardization and Quality Control. This is our local organization of Iraq. We work in it. On this slide, I put an example of the microbiological standard of the food, which you will see the principles 1, 2, 3, 4 in it. This bacteriological standard of meat, chicken, and fish. It is a sample, not the real standard. Type and state of the meat, fresh red meat. In the second column, we will see the microorganisms you must examine. The first test is total bacterial count. The principle number 1. Principle number 2, maximum count per gram. It is permitted in the fresh red meat up to 1 to 5 million total bacterial count per gram, while E. coli, it is permitted from 10 to 50. Salmonella, 0. If you examined the fresh red meat and you cultured it on SS agar to isolate salmonella, and you isolated even one microorganism, it will be a spoiled meat, not acceptable for the consumer, not healthy. It will be a spoiled fresh red meat. The second is minced red meat, also principle number 1, determination of types of microorganism and determination the higher limit of each type. This is the principles to issue a food standard. Total bacterial count up to 1 to 5, that is 10 to 50 million. More than fresh red meat because it's minced meat. Okay? Exposed to higher contamination sources, higher than fresh red meat. E. coli up to 100. Salmonella, 0. You see, this is the principle that for each state of food there is a standard for it. Frozen red meat, we see also Salmonella, 0, while E. coli 10 to 50, total bacterial count just like that of fresh red meat. Frozen chicken, Salmonella, 0, E. coli 10 to 50, while total bacterial count 100,000. This is an example of the milk and milk product. When see a principle of a sixth principle, where for each some product just like milk, there is a grades for each grade, a special standard, grade A, B, C, and the milk powder differ than the liquid milk. The raw milk differs than the pasteurized milk. We must examine total bacterial count and coliform in the milk and milk products, and we classified it to grade A, where the coliform 10 and the total bacterial count of 200,000, where in the pasteurized 330. Here we see the grade B with a higher bacterial content, while C is more than the count in the grade B. The milk powder also, it is permitted up to 90 coliform, and the extra quality about 50,000 total bacterial count per gram. This is per gram, this is per ml for liquid milk. And the standard of the butter. The test you may do is total bacterial count up to 10,000, mold and yeast up to 20. Okay, when you examine a butter and you for mold and yeast and you count it up to 20, it is acceptable. You must examine the butter about cyclophilic bacteria up to 1000, lipolytic bacteria up to 1000, proteolytic up to 50. More than 50, it is unacceptable. Coliform up to 10, and fecal streptococci up to 10 also. So the microbial groups you must examine in the butter sample, food sample, is different than those of milk and different than those of meat, but there is something constant. It is the total bacterial count. It is the first test for each types of foods, but the microbial limit is different from food to another. What about ice cream? The tests are total bacterial count up to 10,000, coliforms up to 10, Staphylococcus aureus up to 10 also, Salmonella 0. You may ask yourself why it is permitted here to found at 10 microbes of Staphylococcus aureus in the ice cream. Isn't pathogenic bacteria, isn't toxigenic bacteria. Yeah, Staphylococcus aureus is a toxigenic microorganism causing staphylococcal food poisoning, and a pathogenic microorganism may cause death, but it is permitted in ice cream to be up to 10 cells per gram because the degree of the ice cream, the temperature is not favorable to produce toxins, so it is permitted up to 10, but higher than 10 the ice cream will be unacceptable. Thank you for listening. I'm available for any questionnaire. We will meet in the Google meeting at our day. Thank you.